Hello, good morning students. Today we'll clear the concept of Southern blotting. Southern blotting is a very important technique for the identification of DNA. Various steps involved in the Southern blotting they are: first, extract and purify DNA from cell. Separate the DNA to be tested from rest of the cellular material in the nucleus. Then incubate the specimen with detergents to promote cell lysis. It frees cellular proteins and DNA. Proteins are removed through organic and non-organic extraction. Then use alcohol precipitation to purify the DNA from the solution. Visible DNA fibers are removed and suspended in the buffer. Second step is DNA fragmentation. Use restriction endonuclease enzyme to break long nucleotide sequence into smaller fragments for purification or identification process. Before appropriate DNA concentration and establishing a restriction digestion with preferable enzyme, keep reagents necessary for digestion process on ice. The components are then added into a PCR tube and mixed by absorbing the contents with the help of pipette, slowly avoiding formation of any bubble. PCR amplifies the number of fragments of DNA obtained from the restriction digest, which are easily separated using gel electrophoresis. Third step, gel electrophoresis. Sort the complex mixture of DNA fragments according to the size. Gels consist of microscopic pores and are solid, may be agarose or polyacrylamide. Nucleic acids are negatively charged and move from left to right. The large molecules are held up while smaller one moves faster, causing separation by size. Gels are stained with ethidium bromide to permit photography under UV light. A single band form is given to intact high quality DNA. Next step is denature the DNA. Now, DNA obtained are double-stranded in nature. The alkalis are used to denature the restriction fragments in the gel that makes double-stranded DNA to become single-stranded. To avoid rehybridization, we use sodium chloride so that DNA is neutralized. Next step is blotting. Transfer DNA from the gel to solid support, that is a carrier membrane. Dry the blot around 80 degrees centigrade or use UV radiation to make it permanent. Next step is hybridization. The membrane bound with DNA are incubated after adding the labeled probe. Usually it requires 1 to 16 hours depending upon the complexity of the probe and the concentration. The probe then binds with the complementary DNA on the membrane with the help of BSA or casing. Then is the washing. Despite using blocking agent, some excess probes bind to the membrane. Wash buffers containing sodium chloride and detergent washes away the excess of the probe. Next step is autoradiography. The particles are exposed to X-ray film when they use radioactive probes or fluorescent probes. If a chromogenic detection method is used, we can see development of color. Next is application. It identifies specific DNA from a DNA sample. Identification of viral as well as the bacterial infection. Important in the study of gene mutation, deletion and rearrangement. DNA fingerprinting, that is maternity and paternity analysis, forensic studies and personal identification. Diagnosis of neonatal and genetic diseases including the cancer. Then is the discovery of RFLP, that is restriction fragment length polymorphism to map crucial genomes. So there are certain advantages and disadvantages of Southern blotting. Advantages, they are less degraded compared with protein and mRNA. As DNA, they are very stable. Effective way to detect specific DNA sequence from large complex sample. Increase sensitivity of fragment detection because of the probe label used for amplifying signals. And the disadvantages, they are not applicable in routine diagnostic setting. It is very time consuming and it also requires large amount of DNA. It does not allow morphologic preservation of tissue, so historic evaluation feature are not available. So in this way, the Southern blotting is there to test, to identify the DNA from the sample. I hope the concept is clear. Thank you.